Hello everyone, can you hear me well? Great. First of all, I would like to thank the organizers for putting together such an amazing event and for giving me the opportunity to present today. And I'm really happy to be back at the in-person CCP-EM Spring Symposium. The last time for me was 5 years ago, so it's really great to be here. And today I will tell you some things about our recent developments in different workflows, and I really hope that there is something for everyone. So let's get started. We will start with cryo-electron tomography and some developments both in sample preparation and on software side. So is cryo-TEM experiencing a revolution? And I think there's obviously many metrics for this. One would be how many cryo-ET talks there are in the CCP-EM. The other one would be looking at the depositions at EMDB. So if we look, wrong button. Yeah, if we look at the depositions from 21 to 22, there's a really large increase, and I think that can also speak for how many labs and researchers are adopting this technique. So let's have a look into the workflow and what cryo-TEM can actually do for you. So because of it becoming more accessible, we can now look at different types of samples, from purified samples all the way to cells and tissues. And in this first part, I would like to give you some examples of work done by Martin and Ben from Abe Koteka's group on individual proteins and virus samples using cryo-TEM. So the first example that I'd like to show is this equine infectious anemia virus-like particles. You can see that this particle is sort of not very round. It has a different curvature in different areas. It lacks the global symmetry. And if you look at multiple of these particles on the grid, all of them are slightly different, which means that they're not really suitable for single particle analysis. So for this work, we collected data on the cryo-SG4 and used multi-particle refinement, which allowed to determine this 2.9 angstrom subtomogram averaging structure of the capsid C6 symmetric hexamer. And here you can see this asymmetric unit colored by local resolution, which extends all the way to 2.4 angstroms. But of course, would this be electron microscopy conference talk without showing everybody their favorite benchmarking sample? So bear with me. I will talk about epoferritin. And the reason for doing this experiment is to really see where our workflow for collecting data and processing data currently is. So if you take away the cellular background and the heterogeneity, like what does this workflow, what can it do? So for this, we collected data on mouse heavy chain epoferritin, and then looked at how much data do we need to go sub 2 angstroms. So the answer to this question was just 10 tilt series, which take only an hour or so, between 1 and 2 hours to acquire, and that led to this 1.8 angstrom map. And then when we expanded this data set to 100 tilt series with 31,000 particles, that allowed us to obtain this 1.5 angstrom epoferritin map. So of course, this is not new in biology terms, but I think it does speak to the maturity of both cryo-TEM data collection and subtomogram averaging workflows, that when sample is not limited, it allows you to go to this high resolution. Okay, so let's change gears and talk about cellular tomography. The cells are much thicker, and we need to make them somehow thinner so we could image them on a TEM. So these 3 instruments are the 3 that are available from Thermo Fisher. We have Aculus 2 on the left. So the workhorse of the workflow, there are over 100 of these dual beams worldwide. And actually, if you look at most of the publications which had used Thermo Fisher dual beam, it will be probably Aculus. Then in the middle, we have a Hydra, which is a plasma fib, and it is really versatile. It can do different applications, including room temperature work. And of course, we have the new kid on the block, Arctis cryoplasma fib, which is really geared towards automation and connectivity. So with that in mind, now I'd like to show you some of the work that was done by Shanjun and Ron Kelly from Abby's group. And the work that I will be showing next was done using Arctis cryoplasma fib. So for those who are not familiar with this dual beam, this diagram summarizes the main features. So it has a CompuStage and an autoloader, which allows you to load 12 grids at the same time. You also have all 3 beams converging on the same area on the sample. So that means that you can measure the fluorescence before, during, and after milling. And it also has a really small chamber compared to the Ieva dual beams, which means that you can run this machine for a long time without contamination. So here's one example grid prepared using Arctis. You can see 47 lamellae that were milled in 48 hours. And just from this one grid, we're able to collect hundreds of tomograms using Thermo 5. If we look at one of the lamellae from that grid, it's one of the thinnest ones here. You can see it's in the middle. You can already sort of see cellular features in there, which makes it quite easy to add your positions for data acquisition. And on the right, we can see a still image from one of the tomograms showing really beautiful ultrastructures. We also wanted to see what statistics we get with this milling procedure of reducing the current every time, like shown on the slide. And we measured the thickness of 100 tomograms and got a mean value of 128 nanometers. So you may be wondering how that compares to Aquilus 2, and we did that comparison as well. So we took 42 tomograms from lamellae that were used, that were milled using CRyo-TEM, and a mean value for those was 172 nanometers. So we were getting thinner lamellae with a plasma fib in this case. So going from statistics, let's have a look into some results. So in this work, we're trying to uncover the chlamydomonas proteome using high throughput visual proteomics. In the middle, you can see a cartoon image of chlamydomonas cell, and on the right, a search map from one of the lamellae. You can already identify so many beautiful features in this search map. And yeah, chlamydomonas have already over 14,000 proteins, but the structural database is quite limited. So thanks to this throughput available using Arctis and Cryos, we were able to collect over 1,500 tilt series from about 150 lamellae, and so far identified 20 different proteins and solved 4 different structures from that data set with more work still ongoing. So in the next couple of slides, I will show you some tomograms from this data set of the sort of things that we saw. So on the left, we can see this really tightly packed pyrenoid full of Rubisco molecules. In the middle, there's a Golgi apparatus visible, and on the right, thylakoids, part of the photosynthetic part of a cell. And then in the next slide, there are 2 examples from mitochondria. So we see all these membranes inside the mitochondria that are decorated with ATP synthase and respiratory complex. And on the right, if we wait for the right slice, there are nuclear pore complexes visible. So in the next slide, I will show you one of the protein structures that we determined from this work. And it's a ribosome structure, 6.2 angstrom ribosome structure from 6 tilt series collected from the same lamella, and in total 9,000 particles. And I think it really speaks about like the quality of the data that we have. But what's really more interesting is that we see significant variability in the ribosomes in this data, and already identified 14 different states. So the next question is, of course, if we map different ribosomes of different states back to the tomograms, what will that tell you? And hopefully that should tell you which protein is doing what, when, and why. So last two slides about sample preparation, and I would like to give some spotlight to Hydra, cryoplasma fib, and you can see the main features on this slide. But the feature that I would like to really focus about is slice and view. So life exists in 3 dimensions, and slice and view allows you to delve really deep into the structure of the cells and tissues without just having to, focus on a single slice or a single lift out. So the way this technique works, that we mill, in this case, 20 nanometer slices using the plasma fib, it takes 6 seconds per slice, and then use SEM to take an image of this surface. And then, so in this work, 335 slices were analyzed that were then used to put together this volume. And so this is a chlamydia cell. It has been only plunge frozen. There's no added stains or fixatives, and you can see like how much detail it actually gives you. So if you take it forward and you render it, you can extract a lot of cellular detail from this analysis. Okay, so let's move forward into the workflow and talk about the data collection. So I think Dan yesterday mentioned about tomography 5 having a lot of new features. So I thought I would summarize some of them here. These 3 features are not too new. So when you're doing tomography, you can add new batch positions with a single click, and you can view all your batch positions on the grid really easily. And of course, you can select different tilt schemes, such as dosimetric. More recently, we also added this improved low assay atlas alignment, which allows you to have these really well-tiled search maps of the lamellae. We also added a lamella angle wizard, which allows you to determine sort of the real zero of the lamella, and integration with TomoLive is now there. Additionally, we now have multi-shot has moved from being able to acquire different positions on the rotation axis to being able to add them off rotation or off tilt axis. So that really allows you to speed up data collection. The numbers, of course, will be different on many different parameters, but in this graph here, we show that with these improvements, each tomogram takes less than 8 minutes to collect. So what really happens when you collect 100 or 150 tilt series overnight and hundreds over the multiple day sessions? Of course, we have to look at all of them and decide what is worth focusing on. And here I thought I would say a few words about TomoLive, which hopefully can ease things for such cases. So what TomoLive does, it actually reconstructs all the tomograms real time as they're being collected by Tomo5 software. And the processing is done on the DMP server, so stuff like motion correction, stack alignment, and then reconstruction happens live as you collect the data, and you can view the results in the browser window, like on the microscope. This is how it looks. It's embedded in Athena, and it's completely parameter free. So it just works in an attended fashion during the recording. The browser window has an integrated 3D tomogram viewer, so you can also check the tomograms, and you can also, besides doing that visually, you can also look at various metrics such as drift, alignment accuracy, and so on. So that our hope is that really this would free up that time which you would spend going over all of your tomograms, and then you can just focus on the ones that show you what you're looking to see. So that brings me to the second part of the talk, and let's discuss some things that help to take single particle analysis from perhaps where it was a few years ago into this sort of high throughput, high resolution single particle analysis that is also really well suited for drug development work. And one instrument that I would like to give some spotlight is Glacios microscope. When I joined Thermo Fisher in 2018, we had the very first Glacios that was ever manufactured, installed in the nanopod. And that was the first microscope that I was using in my new job, collecting data at 24, 25 images per hour. And we really needed to try hard to go close to 3 angstroms on apoferritin. And I think it's just quite fascinating how in the space of 5 years, various different software and hardware improvements are taking us to the new era where it's easy to collect throughput of 650 images per hour, even 1,000 images per hour, depending on your parameters. And as well, we're not counting Glacios anymore on like one hand or two hands. We have over 220 of them in the world. So here's a really short slide about Glacios 2. We were able to push the apoferritin to 1.6 angstroms on the Glacios 2. It has improved stability and optical performance. It also has fringe-free imaging as well as Selectris and Falcon 4 eye combo. And it also has this improved ease of use through automation and Smart EPU. So perhaps you're wondering what Smart EPU is. And this has a little summary of what it consists of. So it's basically an ecosystem of 3 parts. You have the data acquisition that is done through EPU3 that you know, and EPU multigrid. Then it has live evaluation, which is needed for it to make decisions. And that can be done using EQM or embedded CryoSpark Live. And then it also has these decision algorithms, which allow for automated feedback, as well as some smart plugins. So one of the plugins that I would like to highlight, I think many of you will appreciate this, it's called a Smart Selector Filter. So we trained a neural network on a lot of user supplied data to identify good and bad holes. And with a single click, then it looks at the grid square and only leaves the holes that are likely to be good. So it removes holes with no ice or with cracks or with ice contaminations and so on. So I think I really wish this kind of feature was available during the many sessions where I was manually creating 50 or 100 grid squares. And I hope that it'll be useful for people. And one more EPU feature that I would like to highlight that I think is very useful to save time is this automated screening. So everybody knows EPU multigrid, that it can just collect different data sets for you, but you can also use it for screening. So there is this kind of hidden button called fast screening. If you toggle it on, then it goes from selecting every hole in the grid square to just selecting the number that you said. So if you say 5, it'll pick random 5 ones or 1, 3, or 4, and so on. And then with just 2 clicks, you can add all of the grids that are inside a cassette to the queue. And then you have to have a template defined, but then you just click start. And then over the course of perhaps an hour or a bit more, it will go for those grids and go to the grid squares you asked, to these randomly chosen holes, and we'll take images. And then you can look at all of those images on Discovery Viewer in Athena, and that hopefully will save you time again, because you will not have had to sit there for hours manually screening grids, and it will tell you which grid squares from which grids, from which holes give you the best particles. So with this huge increase in speed and automation, we were curious what possibilities this would open for us, especially in the background of drug development. And so the question we asked ourselves, can we solve 12 structures in a single microscope session? So this work was done sort of like intermediate stages of the pandemic, and we worked with Takeda Pharmaceuticals and Utrecht University to see if we could accelerate structural characterization of 12 preclinical stage monoclonal antibodies. So by this point, making spike fab grids was already quite routine, so we knew we could do it. We did do screening on the glaciers by collecting 50 movies and then doing 2D classification as a sanity check. And then for every data set, we collected 1,000 movies on the cryos. And quite surprisingly, all 12 complexes resulted in global reconstructions below 3 angstroms. And we were really surprised by that, because at that point, a lot of papers showing epitope mapping were using 5,000, 6,000, or 7,000 movies as a data set. But what's more impressive is when we locally refined the RBD with the fab bound, and that's only a total mass of about 70 kilodaltons. In all of the cases, we're able to achieve this 3 or low 4 angstrom resolution, and then really accurately define the epitope paratope interface. And having sort of mastered the art of spikes and fabs, we thought, would this also work for much smaller biologics? So for this work, we collaborated with Bicycle Therapeutics here in the UK, and we used EPU multigrid workflow to characterize a number of these bicyclic peptides. And in this slide, I'm showing you just one, which we think is really exciting. It's a biparatopic bicyclic peptide. So it's one that binds to the N-terminal domain and another one that binds to the receptor binding domain attached to each other with linker. And again, using a multigrid approach and having a lot of MNEQ, we're able to solve a lot of structures and define these epitopes. But then we also wondered, if we step away from this high resolution molecular interactions workflow, can we use EPU multigrid to help us with more transient complexes, especially those interesting to the pharmaceutical industry? So the prime example of this are PROTAC mediated complexes, because they're quite transient and they're dynamic. And there we used EPU multigrid to screen dozens and dozens of conditions, usually just 500 or 1,000 movies at a time, to come up with the conditions that allowed us to visualize the whole ternary complex. So here you can see the results from this work, a complex of DDB1 cereblon attached to the ER ligand binding domain via this PROTAC mediated proximity. And you can read more about this work on the Arvinas website. And last but not least, I would like to talk very briefly about this amazing CDK activating kinase story that was published by Basil Gareva's lab just a few weeks ago now or by archive. So for this work, again, EPU multigrid and Glacier's cryos workflow was essential to just determine a really large amount of structures. So initially, CAT complex in complex with 14 different inhibitors in duplicate was screened on the glaciers, and just 1 hour data collections allowed us to get 3.5 and 4.5 angstrom structures, and all of that would already tell whether we have good orientation distribution and whether the ligand is bound. And then, of course, those data sets could then be increased in time. And in this case, on the glaciers, it was possible to go all the way to 2.3 angstroms using 22 hours of data collection. And of course, all the best grids and the compounds went to the cryos to go sub 2 angstroms. So with that, I would like to finish my talk. I would like to acknowledge everybody involved in various parts of the story. I would like to thank you all for listening, and I'm happy to take questions.